pathway and a unique post-translational modification known as hedgehog autoprocessing. As a brief background, in my time in the program, I've been in the PhD in chemistry program for about 9 semesters. I did my prelim in January 2020. I have about 6 publications and about as many presentations and posters. So to begin with, I'd like to give just a little bit of background on the hedgehog signaling pathway, its role in the body, and why we study it. So hedgehog signaling is necessary for proper body development, development of symmetry of your brain, eyes, and digits on your fingers, as well as regulation in later life in adult stem cell proliferation. The naming convention for hedgehog is a bit interesting in that hedgehog genes are found in more than just hedgehogs. They can be found in organisms as simple as a Drosophila, a fruit fly, all the way up to complex organisms such as humans. The discovery of the hedgehog genes originates from a study of fruit fly larvae, where they found that the wild type larvae had these distinct tentacle shaped objects that look like bands. However, when a particular gene set was mutated, they found that the morphology of these bands shifted to that resembling more of a lawn, which they felt resembled the quills of a hedgehog, and thus the naming convention stuck. Now, hedgehog signaling is important to study therapeutically as it's been linked to a number of disease states. When the signaling pathway is inhibited, typically congenital malformations can form, most of which, or the most studied at least, being holoprosencephaly, which is hallmarked by a failure of the body to develop proper symmetry. One of the most well-known cases is an interesting story from, I believe, the 1950s, which is the story of the one-eyed sheep. Here, a flock of sheep had birthed cycloptic calves. After studying the environment that the mother sheep were feeding or grazing on, they found that the plants that the mother sheep had been ingesting contained a natural product sterile-like molecule known as cyclopamine, which is a potent inhibitor of the hedgehog signaling pathway and aptly named because it can cause cyclopia in the calves. On the other side, if hedgehog is overactive or overexpressed, this has been linked to a number of cancers, including prostate, bladder, and breast, mostly in later life. Now, the signaling pathway is very complex and dynamic, and for the most part, the signaling pathway itself is beyond what my research specifically focused on. However, I feel this slide will give you some important context as to why I studied what I did and just some appreciation for the signaling pathway itself. So if we begin with the signaling cell, the pathway begins with the translation of a 2-domain precursor protein that undergoes an autocatalytic self-degradation utilizing a molecule of cholesterol. The precursor is divided into its 2 domains. The red domain indicated here, we call HHN, is responsible for downstream signaling. HHN is covalently linked to a molecule of cholesterol during its autoprocessing event, and it's also N-terminally modified by a molecule of palmitate before being secreted from the signaling cell as the mature signaling ligand shown here. In the receiving cell, there are 2 transmembrane proteins, first being patched and the second being called smoothened. In the native state, patched acts as an inhibitor over smoothened, preventing further downstream signaling. However, when the hedgehog ligand associates with patched, the inhibition on smoothened is relieved, allowing the signaling cascade to continue, and the hedgehog genes are further activated. So overall, this system is very complex, and it's actually even more than I show here, but I just wanted to call attention to the fact that I study this initial interaction that is a gatekeeper over the entire signaling pathway. So if we take a closer look at hedgehog autoprocessing and look at it from a chemist's eye, the 2 domains of the precursor protein are linked by a conserved glycine-cysteine pairing. The HHN domain, which is necessary for downstream signaling, plays a bystander role to the actual autoprocessing event. Its identity can actually be swapped out for other proteins, such as fluorescent proteins, or even as small as like a histidine tag. All of the catalytic machinery for autoprocessing is housed within the HHC domain. Two conserved residues that I've indicated here will be important later in my talk. The first being cysteine 1 at the first position of HHC, and the second being an aspartate at the 46th position. In the native state of the precursor protein before any autoprocessing occurs, these two residues have their side chains in a coupled state through a hydrogen bond. This retains the thiolate of C1 in an inactive form, reducing premature activation of the precursor. Upon recruitment of a molecule of cholesterol, which is the natural substrate for HHC, the thiolate of C1 is freed and is allowed to attack the preceding carbonyl of a glycine residue. The peptide backbone is then rearranged following N to S acyl shift, leaving us with a thioester intermediate of our peptide linkage. Here, D46 plays its second key role as a general base catalyst. The carboxylate group of the aspartate assists in deprotonating the hydroxyl of cholesterol, which then is a suitable nucleophile to attack the carbonyl of our thioester. Upon collapse of the tetrahedral intermediate, the two domains of the precursor are cleaved from one another. Cholesterol is esterified through a covalent linkage to the C terminus of HHN and proceeds for downstream signaling. Now, the autoprocessing reaction could be a target for drug design, as it's necessary for effective signaling to occur. It's early in the signaling pathway. As I showed, it's one of the first interactions of hedgehog signaling and acts as a gatekeeper. And third, the chemistry of hedgehog proteins appears unique to hedgehog proteins. However, development of therapeutics for this drug target are impaired due to a lack of complete understanding between substrate structure and enzyme activity. To date, there hasn't been a complete crystal structure of the full HHC domain. So we're left with basically structure activity assays to better understand the interaction between substrate and active site. So my research approaches this lack of understanding by investigating a panel of sterile analogs to better understand the substrate scope of HHC. Some previous publications have shown that sterols beyond cholesterol can serve as potential nucleophiles or potential substrates for HHC, though there hasn't been a very in-depth look at the limitations of substrate tolerance. The second aim of mine has been to use synthetic sterols to rescue an otherwise inactive form of HHC. And finally, though I won't have really a chance to talk about it today, I developed a novel cell reporter system for studying human protein in a high throughput system. So in order to approach the first topic of my research, which is to better understand the limitations of HHC substrate tolerance, we needed to take a look at the native substrate, which is cholesterol. And through a rational deconstruction, we broke cholesterol into three distinct domains that we could target for modification: the isooctyl tail region, the A ring of the tetracyclic ring system, and the hydroxyl group which acts as a nucleophile during catalysis. So these are the three sites of cholesterol that we look to make modifications to really ask the question, what are the limits of HHC's substrate tolerance? In order to measure and quantify these alternative substrates, we needed a tool that was both quantitative and modular. So we looked to a previous fret-based optical assay that a now graduated graduate student from our lab had developed named Tim Owen. What Tim had done was take the catalytic domain, or the HHC domain, of Drosophila and fuse it between two fluorescent pairs, CFP and YFP. This precursor protein, which we call CHY, is able to undergo what's known as FRET. What FRET is an energy transference between the two fluorescent proteins. Following excitation at 400 nanometer light, we can detect emission from YFP at 540. However, this energy transference is strictly dependent on the space between the two fluorescent pairs. If we were to, say, cleave CFP away from the HY domain following hedgehog autoprocessing, CFP is sterylated by cholesterol. However, now the distance between our two FRET partners is too great for the energy transference to occur, and we lose our emission at 540. In practice, what this looks like is what we call FRET traces, and I'll show you many of these today. It's one of our predominant tools in measuring hedgehog activity. The top trace here represents our intact CHY precursor, which has a stable signal. However, following the addition of cholesterol, CFP is cleaved from HY, and no longer will we see the transference of energy from CFP to YFP. We lose our emission at 540, and we lose our FRET signal. We often like to validate our FRET results by SDS page as a secondary assay. So here, again, we're using the CHY protein. In the absence of cholesterol, CHY runs predominantly in its precursor form. However, with the addition of cholesterol, we see a loss of precursor and generation of our product HY and CFP bands. So these are two of the main tools that I'll be discussing today. So we then look to test some tail-modified sterols within wild-type HHC. We somewhat serendipitously stumbled upon abiraterone and galetterone. We were actually testing a chemical library for potential inhibitors of cholesterolysis or hedgehog autoprocessing, and we found these two sterol analogs that appear to activate hedgehog activity. What's important to note is that the hydroxyl group and the ring system of these sterols essentially overlap with cholesterol. All the difference structurally exists within the tail area. So if we look at our FRET analysis of these sterol analogs, again, CHY with no substrate added gives us a stable signal. As a positive control, cholesterol is added, and we see a loss of FRET over time. And with addition of these sterol analogs, abiraterone in green and galetterone in blue, we see FRET loss, suggesting that these are acting as alternative substrates for HHC. And based on their rate of catalysis, it initially looks like HHC does not discriminate tail-modified sterols from cholesterol. We then looked at these sterols in a titration-based experiment where we titrated the concentration of sterol and measured the rate of initial velocity. After plotting in a Michaelis-Menten plot, we identified their binding, the binding affinity of the sterols for HHC's active site. This is measured or represented by the Km value, which is a measurement of binding affinity. Remember that a lower Km, the tighter binding between enzyme and substrate complex. So here, cholesterol has a Km of 1 micromolar and abiraterone of 12. You could consider this as abiraterone binds 12 times weaker relative to cholesterol. But a better indicator is what's known as catalytic efficiency, which is considering both the rate of catalysis and the binding affinity of substrate and enzyme. Here we see that abiraterone is about within tenfold of cholesterol, and galetterone actually appears improved efficiency over cholesterol, which was surprising, as this was one of our first sterol analogs that behaved apparently better than cholesterol. So we then validated our FRET results by SDS page. Here we're using a chimeric fusion protein that has the sonic human HHN domain fused to Drosophila HHC. When this protein is reacted with cholesterol, we see a time-dependent loss of precursor protein and generation of our product peptides. We see similar results with abiraterone and with galeterone. What's important to note is that the rate of loss of precursor for abiraterone and galeterone matches what we saw by FRET, where abiraterone seems a little bit slower than cholesterol and galeterone appears faster. So this is good support of our FRET data. And it's really suggesting that these are viable alternative substrates. We then wanted to validate that the same catalytic mechanism was occurring and that we were forming a covalent conjugate between sterol and N-terminal protein. So we assayed this by reverse phase chromatography, which is a denaturing mechanism for investigating protein reactions. Here we look at the retention time of our protein sterol complex. Free HHN has an earlier retention time to cholesterylated HHN. This is due to the cholesterol molecule adding some hydrophobic character to the protein sterol complex, increasing its retention time over the column. So if we then compare our two sterol analogs, we see that abiraterone and galeterone conjugated to HHN run similar to cholesterol, which is a good indicator that we're forming a covalent linkage between protein and sterol. So then the next obvious question was whether these HHN non-canonical sterol conjugates could service as ligands for downstream hedgehog signaling. Now, this experiment was conducted by a previous undergraduate known as, or by the name of Brandon Bordeaux. What Brandon did was titrate HHN conjugated protein into cultured mammalian cells that is sensitive to hedgehog signaling. And after measuring the degree of hedgehog signaling, he plotted in a dose response experiment that of cholesterol conjugated HHN, galeterone, and abiraterone conjugated HHN. And he found that their effective concentration for activating downstream signaling was essentially on par with one another. They had an EC50 between 1 and 3 nanomolar. So this suggests that these non-canonical HHN conjugates can service as potential activators of downstream hedgehog signaling. This is therapeutically significant because abiraterone is actually a marketed FDA-approved medication for late-stage prostate cancer. What abiraterone is intended to do is inhibit the activity of an androgen synthesis enzyme known as CYP17A1, effectively turning off one oncogenic pathway. However, what may be occurring is that while we may be inhibiting CYP17A1, abiraterone may have a polypharmacology and activate hedgehog signaling unintentionally. In other words, you're turning off one cancer pathway, but accidentally turning on another. So this just gives some context as to why it's important to study the substrate scope of HHC beyond the native substrate cholesterol. We then shifted our focus away from the tail region of cholesterol and started to make modifications to the A ring of the tetracyclic ring system. All the sterols that I'll present after this point were synthesized by our collaborators at SUNY SF and Dr. Heiner's lab. What Dr. Heiner and his group prepared was first an A-ring truncated sterol that has the A-ring reduced from a 6-membered to a 5-membered ring and has the hydroxyl group adjusted from a secondary to a primary alcohol. They also prepared an A-ring expanded sterol that has an additional ring system. It's now pentacyclic, again with a primary alcohol. And finally, they prepared actually a natural product sterol known as caprostanol that has a bent ring system. We then tested the activity of these sterols by FRET and found that all 3 of them are tolerated by HHC, though to varying degrees. While the truncated sterol behaved apparently near on par with cholesterol, the expanded and the bent ring sterol were where we began to see some limitations in substrate tolerance of hedgehog. So we plotted these in a titration-based experiment, titrating the concentration of sterol and calculating the initial loss of fret. And here's where we really began to see differences in substrate tolerance, especially by the expanded and the bent ring sterols. So although HHC will tolerate sterols beyond cholesterol, that tolerance is not unlimited. So if we do a little bit of a recap of this sterile analogs that I've shown so far, abiraterone and galerone were relatively well tolerated by HHC, especially galerone. This is a relative catalytic efficiency to cholesterol, comparing the activity of these sterols to cholesterol. If we then look at the A-ring modified sterols, the A-ring truncated is a suitable substrate, but the expanded had only 1% activity relative to cholesterol. The caprostanol is actually less than 1%. So this led us to the question, can we make modifications within the enzyme active site that would improve the substrate scope of HHC? Essentially, can we make a better enzyme that would facilitate catalysis with caprostanol more effectively? So we thought about where to make a mutagenic change, and we came back to the 46 position within HHC. In wild-type protein, this is an aspartate residue, and it acts as a general base during catalysis. So we thought to swap to histidine, which in theory can also act as a general base. Now, often in literature publications, a D to H mutation limits, if not fully abolishes, enzymatic activity. So we weren't fully sure whether this would work or not, whether a D46H mutant would process the native substrate cholesterol or expand substrate scope. So we first tested with cholesterol, and we found that not only does D46H process cholesterol, it does so on par with wild-type protein. We then followed this by SDS Page using a fluorescently labeled cholesterol analog, and we detected covalently conjugated N-terminal protein, suggesting that D46H follows a similar catalytic mechanism to the wild-type enzyme. So this is good. We have a potentially, or a mutant that has potential for expanded substrate scope. So then we tested a panel of sterile analogs. And there's a lot of data on this slide, so really what I want you to focus on are these two sterols here, caprostanol and epicaprostanol. If we look at their structure in 3 dimensions, cholesterol is represented in blue. Caprostanol with the bent ring system has its A ring displaced about 90 degrees. And epicaprostanol has the hydroxyl group in an epimeric form compared to caprostanol. So we've already established that with wild-type protein, caprostanol is a weak substrate. However, it appears to readily process with D46H. In fact, D46H has a 200% preference over wild-type for this bent ring sterol. Perhaps even more interestingly is that epicaprostanol, which has no detectable reactivity with wild-type, we could detect processing with D46H. So it seems like this is a mutation that expands the substrate scope of HHC. The other sterols that are shown on this table have either a comparable, if not a preference for D46H over the wild-type form of the protein. So that leads us to the obvious question of, well, what can D46H do that the wild-type protein can't? What's special about D46H? So what I want you to focus on really right now is just the center of the slide here. What we rationalized the enhanced substrate scope of D46H to be a result of is the differences in transition state stabilization between the two forms of the protein. In wild-type protein, shown here, reacting with native substrate cholesterol, the hydroxyl of cholesterol attacks the carbonyl of our thioester, and that tetrahedral intermediate is stabilized by the aspartate at the 46 position. However, if caprostanol was in the active site, that bent ring system may displace the aspartate in such a way that it's now too far away for a hydrogen bond to stabilize the intermediate. On the other hand, a histidine at this position is able to not only stabilize through a hydrogen bond, but the imidazolium group can stabilize through electrostatic interactions. Now, electrostatic interactions are more long range and less geometrically constrained relative to a hydrogen bond. So despite caprostanol displacing the active site, that can be compensated for by the more long range electrostatic interaction between the functional group of histidine and our intermediate. So we then thought to make a different mutant where instead of doing a general base swap, we mutated out the aspartate to an alanine, basically removing the general base activity of our active site. Now, when the aspartate is removed, there's no longer general base catalysis acting upon the cholesterol hydroxyl group, and we essentially see no reactivity of cholesterol with a D46A mutant protein. If we visualize this by FRET, we see no change in FRET signal with D46A and cholesterol. And by gel, we see no generation of our product bands with a D46A. So we can say that a aspartate to an alanine mutation completely abolishes the activity of cholesterol. However, we then began to make modifications to the nucleophile, to the hydroxyl group of cholesterol. And a seemingly simple alteration of expanding the hydroxyl by one oxygen group, transitioning from a hydroxyl to a peroxyl functional group, we surprisingly found rescue of our otherwise inert D46A mutant protein. We see by FRET and by gel that there appears to be rescued activity. What we rationalize this as to be a result of is what's known as the alpha effect, where your nucleophilic atom has an electronegative atom adjacent or alpha to it that's able to increase the nucleophilicity of your attacking atom relative to its pKa. Essentially, you have a more or a hypernucleophilic attacking atom. So we then wondered whether chemical rescue by general base bypass of D46A is unique to peroxysterols, or would other alpha effect sterols behave similarly? So the Heiner group prepared an aminoxy sterol derivative, which is an alpha effect, and we see reactivity of D46A and wild-type protein. And on the flip side, they switched the atoms and made a hydroxylamine alpha effect substrate, again processing D46A and the wild-type protein. So this is evidence that it's not just limited to peroxides. There's actually a whole family of hypernucleophilic sterols utilizing the alpha effect that can rescue the activity of D46A. We also attempted to rescue catalytic activity through a more conventional means through substrate-assisted catalysis. Essentially, what we tried to do was take the general base activity of the enzyme that's now missing and put it directly onto the substrate molecule. So here we're showing cholesterol reacting with D46A. Again, it's an inactive substrate. However, perhaps most interestingly out of this panel was that a split ring diol recovered activity of D46A. We've thought about this, and the most reasonable explanation we could think of was that one hydroxyl group was pulling the hydrogen away from another and sort of acting as an intermolecular general base to facilitate catalysis. So we then tested this hypothesis by putting the functional group of aspartate, the carboxylate, directly onto our molecule at the 2 position. And we found chemical rescue of D46A using basically a wild-type mimetic sterol substrate. We then tried to mimic D46H by putting the imidazole functional group at the 2 position. And again, we found chemical rescue. And then finally, just as a proof of concept, we tested a picoloyl sterol, just showing that you need a hydrogen bond acceptor in this vicinity for intermolecular general base catalysis to occur. In a more schematic way to explain this, again, cholesterol is inactive due to the lack of an aspartate acting as a general base. However, if that aspartate carboxylate group is added directly onto our substrate sterol, now we have intermolecular general base facilitating catalysis. So, in summary, my research has focused on investigating the substrate scope of the hedgehog precursor protein. I initially began by studying tail-modified sterols, and I found that these were readily accepted by HHC. And abiraterone specifically is potentially demonstrating polypharmacology and could be activating an oncogenic pathway accidentally. I then shifted focus to A-ring sterols, or A-ring modified sterols, and determined that this is where we began to see limitations to substrate tolerance, specifically in the bent ring caprostanol molecule, which had a less than 1% activity relative to cholesterol, at least with wild type protein. However, we were able to make a more substrate accepting mutation D46H protein that enhanced the catalytic efficiency of caprostanol by 200%. And then finally, I investigated some nucleophile altered sterols and found that there are two classes of sterol analog that can rescue the activity of an otherwise inactive form of hedgehog. One family utilizes the alpha effect to bypass the need of a general base at that position, and the other family utilizes intermolecular general base catalysis to sort of mimic the activity of the wild type enzyme. I would just like to acknowledge my research group. Andrew Zihan and Jayu have been my buddies all the way through. Alex was a previous research technician who has since taught me everything I know about cell culture. Rebecca really did a lot of the foundational work for our cell culture project, which I didn't really get a chance to talk about today. Brandon did a lot of the cell culture work on the tail modified sterol project, and Dr. Callahan has been an excellent mentor over the years, and I couldn't ask for a better PI. Collaborators, Dr. Heiner and his lab prepared almost all of the sterols that I showed today. They're really masters in synthetic sterol chemistry. Dr. Wong at RPI did a lot of our mass spec and NMR. Dr. Inglese at NCATS is associated with our cell project, and Dr. Bonavalli did a lot of our modeling work. And finally, I'd just like to thank the committee, who've all been excellent mentors to me. And finally, I'd just like to thank the audience for your patience and time, and I would be more than happy to take any questions.
Thank you very much, Dan, for an excellent presentation.
Thank you.
Now is the time for the general audience to ask your questions.
Yeah, Dan, I actually had a question for you.
Yep.
Specifically about the alpha effect sterols, the hypernucleophilic sterols.
Okay.
So, let's see. I definitely understand what you're saying. I think this is really, really well thought out experiments. The only thing that I think is a little bit weird for me is that by introducing the second atom, I understand that the oxygen, the OH, becomes much more nucleophilic, but you also change the distance between the attacking nucleophile and the hydrocarbon ring of your sterol. Did you guys consider that as a potential factor in why you're seeing these work better than traditional sterols?
That's a good question, actually. As part of my research, I've tested probably 400 sterol analogs, and we have thought of just extending out the hydroxyl by 1 or 2 or 3, putting a PEG linker there, and those did not rescue D46A. It seemed necessary for some form of alpha effect functional group to be at that 3 position. It's not just a distance result.
Right. Hey, all right, thanks, Dan.
Okay, any other questions? All right, if there are no other questions, then let's thank Dan again. And I would like to ask the general audience, please, to leave the meeting.
Great job, Dan.
Thanks, Chris. Thank everyone.